students, myself Dr. Pallavi Rai Lavale, and I am going to teach you the unit 3 of BP 405T. We are going to study about the unit in the unit 3, the nutritional requirements of plant tissue culture. So, basically what is the requirement, what are the functions of the plant tissue culture or the nutrient medium of the plant tissue culture. It has to, the nutrient medium should be enough to provide water, mineral nutritional needs, then the vitamins, the plant growth regulators, proper atmospheric gases, as well as it should also be able to remove the plant metabolites produced inside the in the culture, which is generally the waste material. So, a good nutrient medium should have all these qualities. So, you just understand that like when a plant grows, when the plant is growing, it requires various nutritions and which we provide through the soil. So, basic requirements are what? It requires a carbon source, which normally the plant gets through the carbon dioxide, it gets water, it gets the minerals, and after that the plant is itself so you can say competent that it starts synthesizing various metabolites from the glucose which it has synthesized and the messages which it has from the DNA. It starts synthesizing newer and newer enzymes, and the processes go on gradually. From the glucose itself we have the development of different types of the plant the secondary metabolites. So, all the sources it is getting directly either through the air or through the soil, or the minerals are also there in the soil. Now, we are going to do this whole technique inside the lab. So, in the lab also we have to understand that we have to provide all these things if we want that the plant should grow at a good rate. Okay. So, here first of all we have to give a carbon source and then we have to give a nitrogen source. These are the, I would say that they are the macro requirements, macro elements required by the plant in a very great amount, for as they are the basic building blocks of the plant. So, when we talk about the carbon source, so carbon source generally comes in the form of sucrose, fructose, lactose, starch and many more. You know, there are many sources of carbon. So, we can use all of the sugars, either monosaccharides or polysaccharides, as source of carbon. Then we have nitrogen as the source. Nitrogen source again is very important. So, where are the places where nitrogen is involved? Nitrogen is present in all the amino acids, it is present in the DNA, it is present in the enzymes. Okay. So, nitrogen you can see is present in almost all the parts of the plant. So, again it is required in a great amount, and because nitrogen cannot be taken directly from the atmosphere, nitrogen has generally is sent to the plant through the roots. Okay. So, in the lab we can add nitrogen again in two different methods. First is in the defined form, second is in the undefined form. Defined form means where we are adding it in the form of major inorganic ions. Okay. We are putting any kind of nitrate, any nitrate salt. If we are adding any nitrate salt, it is a type of a inorganic salt which we are giving to the plant. So, it is defined. We know that we are going to give say 5 microgram of the nitrate, we are adding to the medium, we know the concentration. Okay. Then second is the minor amino acids. Yeah, amino acids are also a source of the nitrogen. So, if we are adding any kind of amino acids like we take histidine, okay, histidine has some nitrogen. So, we again have a, we know how much amount of nitrogen is going inside the plant tissue culture. So, that is why these are the defined types of nitrogen sources. In undefined, for example, if we are using any kind of naturally nutritive agents, like we are using the coconut milk, we are using yeasts, we are using corn, Okay. So, these are the natural agents. We have soybean casein digest, something like that. If we are using such kind of nutrients, then in that case we do not know that how much amount of nitrogen is present. So, that comes under the undefined type of the nitrogen source. So, further we will move to the different types of the nutrients. So, in the last diagram we got the idea that the two main sources, that is carbon and nitrogen, how they can enter the plant in the plant tissue culture. Here we will be discussing one by one that what is the composition of a culture media. First of all, it should contain the inorganic nutrients. In inorganic nutrients, first they are the macronutrients like nitrogen. As I told, nitrogen is a very important part of all the enzymes, amino acids, DNA, etc. Then phosphorus. Again, phosphorus is also a very important part of the DNA. Okay, it is present in the DNA structure, it is present in the ATP, it is present in the NADP. So, phosphorus again it is also present in the phospholipid bilayer of the cells. So, phosphorus is present at many places in the inside the plant, and again it comes under the macronutrients. Then we have the potassium, calcium. Now, many of the processes like in humans, in animals, we know about the sodium potassium pump. Same way, potassium is involved in major processes of osmosis, diffusion, etc. Then calcium. Calcium is the building block of the cell wall of the of the plant tissue cells. Okay. So, the cell wall it is composed from calcium. Then we have micronutrients like boron, copper, iron, manganese and zinc, which are also required. I would like to mention here again, which I have missed, that magnesium it also comes under a macronutrient, because magnesium is the main ingredient of the chlorophyll. So, chlorophyll which is responsible for the synthesis of glucose, it basically traps the sunlight, it is containing magnesium. So, magnesium again is a type of a macronutrient which these plant cells require. Then we have certain organic nutrients like vitamins, like vitamin B1, B6, B3, B5. Then amino acids like L-arginine, asparagine, cysteine, hydrochloride, glutamine. Carbon sources like glucose or maltose. Growth hormones like auxins, cytokinins, gibberellins, ethylene and abscisic acid. So, these are again required as a main part, you can say, composition of the culture medium. So, we have inorganic molecules and then we have the organic molecules. Other than that, many times the things which are added are the protein hydrolysates, yeast extracts, fruit extracts, coconut milk, solidifying agents like agar, alginate, gelatin, iron source, EDTA and antibiotics. pH of the medium should be in a range of 5.6 to 6 before autoclaving the culture medium. So, once we have added the inorganic materials, the organic nutrients and the other substances, after mixing all, we will maintain the pH of the medium. So, pH of the medium has to be maintained before autoclaving, because after autoclaving it becomes difficult and we cannot avoid any kind of contamination if the pH is measured after doing the autoclaving. Okay. So, it should be in the range of 5.6 to 6, which is generally preferred. However, depending upon which type of medium is mentioned or which we are following, there may be certain variations. This is a standard pH which I am mentioning here. Now, if we look one by one regarding these different types of inorganic nutrients, generally mineral elements they play a very important role in the growth of plant, and basically the function of nutrients in plant growth is extensive. Around 15 elements they are very important in the whole plant, and in general I am talking about in general around 15 elements have been identified, and they are necessary that when we are doing the plant tissue culture we have to provide these nutrients to the particular plants. The macronutrients, basically generally here the concentration which we provides, they are required in a concentration of greater than 0.5 millimole per liter. They are known as the macronutrients, and they mainly are what? They are nitrogen, potassium, phosphorus, calcium, magnesium and sulfur. Nitrogen, it is required in the range of 2 to 20 millimole per liter, and it influences plant growth rate, essential in plant nucleic acids, proteins, chlorophyll, amino acids and hormones. So, it has major role at all these places. Phosphorus is required in a concentration of 1 to 3 millimole per liter, and it is abundant basically in the meristematic tissue, fast growing tissue and essential in the photosynthetic respiration as well. Potassium, it is required in a higher amount, about 20 to 30 millimole per liter, and it is necessary for cell division. Meristematic tissue helps in the pathways for carbohydrates, proteins and chlorophyll synthesis. Calcium is required in about 1 to 3 millimole per liter, and it is involved in the formation of cell walls and root and leaf development. They participate in translocation of sugars, amino acids and ties up the oxalic acid, which is a toxin. Magnesium, it is required in the concentration of 1 to 3 millimole per liter. It is involved in photosynthetic and respiration system. It is active in uptake of phosphate and translocation of phosphate and starches. Sulfur is required in the concentration of 1 to 3 millimole per liter, and it is involved in the formation of nodules and chlorophyll synthesis. So, these are all those minerals which are required in a concentration of more than 0.5 millimole per liter, and they all have a very active role in different processes inside the plant cells. Now, we move to the next topic, that is the micronutrients which are there. So, as a definition of macronutrients was there, the micronutrients are those nutrients which are required in a concentration less than 0.5 millimole per liter. Okay. So, they are required in a lesser amount. Overall, the plant thrives on 17 elements, out of which 4 like carbon, hydrogen, oxygen and nitrogen are derived from the atmosphere, and the rest 13 microelements like boron, copper, iron, manganese, zinc, cobalt, molybdenum, nickel, aluminium, iodine, ferrous, sodium and chlorine. These are the nutrients which are generally you have to give them through the soil in the form of fertilizers or manure when we are doing any kind of farming. But here it is plant tissue culture. So, we have to add through different types of salts. We have to take different types of salts and we have to add those in the nutrient medium. Now, depending upon what is our aim, whether we want to do any kind of root culture or only shoot culture or flower culture, we can make changes in these micro elements and accordingly either increase or decrease their concentration depending upon our aim. A media lacking in these micronutrients does not support healthy and wholesome growth, and the plant exhibits deficiency symptoms such as pigmentation, absence of vessels, presence of narrow cambial zone, cellular hypertrophy and symptoms of chlorosis due to absence of ferrous and sulfur. Now, if we look at the micronutrients, their concentrations and their role in the plant tissue culture. So, we start with iron, which is required in amount of 1 millimole per liter. It is involved in cell division, respiration, chlorophyll synthesis and photosynthesis, and generally it is added in the form of ferric sodium aditate, because it is the sodium salt of EDTA, because EDTA generally chelates with many other ingredients, and iron is also very reactive. So, it is generally along with EDTA, the ferrous remains retained in its intact form and can be utilized by the plant tissue cultures. Manganese, which is there in the concentration of 20 to 90 millimole per liter, it is involved in cell elongation, regulation of enzymes and growth hormones. It also assists in photosynthesis and respiration. Boron is required in a concentration of 2 to 5 millimole per liter. It is responsible for cell division and cell elongation. Copper in the concentration of 0.1 millimole per liter is required for various processes as it acts as a cofactor. Same is the case with molybdenum, which is required in a concentration of 5 millimole per liter, and it is also involved as a cofactor. Cobalt and zinc are also important cofactors. Cobalt is required in a concentration of 0.1 to 1 millimole per liter. Zinc is required in a concentration of 1.5 to 30 millimole per liter. Other than that, iron, nickel, aluminium, chlorine, sodium, they are also required, and they have their own processes and concentrations which have been well studied and well reported. In organic nutrients, generally we add for vitamins, amino acids, we also have certain growth regulators which are added inside the medium, and they help in maintaining the concentration of nitrogen. Okay. So, most culture plants are capable of synthesizing essential vitamins, but not in sufficient amount. To achieve the best growth, it is essential to supplement the tissue culture medium with one or more vitamins and amino acids. So, we should not just fix to the inorganic materials, we have to add certain organic materials in the form of vitamins also. For example, thiamine, which is vitamin B1, here it is an essential coenzyme in the citric acid cycle, and it is required mostly in the tissue culture and is considered to be essential. Nicotinic acid, which is vitamin B3, it is required in a concentration of 0.5 milligram per liter. Pyridoxine, vitamin B6, required in the concentration of 0.5 milligram per liter. Calcium pantothenate, which is vitamin B5, it is required in 0.1 milligram per liter, are known to improve the growth of the tissue culture materials. Myoinositol, which is a part of the B complex, is in phosphate form, is a part of the cell membranes, organelles, and not essential to growth, but beneficial, basically have an important role in many of the biosynthetic pathways. So, when you have to perform any secondary metabolite synthesis, there the role of myoinositol comes into picture. Then you have many more important vitamins like cyanocobalamin, which is vitamin B12, riboflavin, vitamin B1, folic acid, which is vitamin M. Then you have 0.5 milligram per liter concentration for folic acid is required. Then you have biotin, para-aminobenzoic acid, ascorbic acid, alpha tocopherol. These are also added in special cases, but their exact role is yet to be understood. So, depending upon the culture, whichever culture you have following, you have to see if it is mentioned that vitamin E has to be given. So, generally if we are adding any tocopherols or vitamin E, they have the role in synthesis of steroidal compounds. So, if your aim is to produce certain drugs which are rich in the steroidal drugs, like for example diosgenin, so there you may have to give more source of alpha tocopherol, because it will automatically enhance the synthesis of the diosgenin. Now, if we talk about the amino acids, so there are different types of amino acids. You must have studied like the essential amino acids and the non-essential amino acids. So, many plants are able to synthesize their own amino acids. They come under the non-essential amino acids, but when they are not able to synthesize, at that time we will call them the essential amino acids. The most common sources of organic nitrogen used in the culture medium are the amino acids. Example, casein hydrolysate. L-glutamine, L-asparagine, arginine, methionine and adenine are generally used. When amino acids are added alone, they can be inhibitory to the cell growth. So, we should not add them alone. Tyrosine has been used to stimulate morphogenesis in cell cultures, but should only be used in agar medium. We should generally have a agar medium. We cannot use it in a liquid medium because it generally promotes the shoot growth. Supplementation of cell culture with adenine sulfate can stimulate the growth and greatly enhance shoot formation. For carbon source, we have discussed it in detail already. Generally, the preferred things are either starch or sucrose in the concentration of 2 to 5% or glucose or fructose can be used for the good health. Even maltose and raffinose can also be used. And generally, if we are using any you can say chemical type of carbohydrate source, we can go for sucrose, dextrose, glucose etc., or we can go for mannose, sorbitol, pentose, sugar alcohols, glycols etc. But there are other sources also, like we can go for the potato starch, we can go for rice starch, we can go for honey as a source, okay. We can use pectin as a source from certain fruits. If we are isolating, we can use cellulose from certain plants as a source. So, that can also be used as a source of the carbohydrates. Now comes a very important part, that is the growth hormones and the growth regulators or the modulators. See, generally we use regulators or the modulators term because it is not necessary that always the growth has to take place at a continuous process. There has to be a balance, there should be good growth as well as there should be a check on the growth also. So, we will generally call it a growth modulator. So, whenever we are doing any plant tissue culture, the success basically depends on the amount of the plant hormone which we are using. So, a good biotechnologist will always have a balance of which particular plant hormone has to be chosen and in how much concentration it has to be chosen. Generally, we have 5 different types of the plant growth regulators. Auxins generally they produce the cell elongation, cell division, okay, and they are generally they prefer the growth of the root culture, okay. So, auxins are used there. Ethylene is generally responsible for ripening of the fruits. So, generally if we are doing any kind of fruit culture, we will prefer ethylene. Abscisic acid is generally responsible for the dormancy of the plants or senility of the leaves. So, generally it is used when we want to stop the further growth of the cells. Cytokinins are generally responsible for the lateral growths of the shoots as well as the roots. So, if we are using any kind of leaf culture or lateral roots or lateral branching culture, at that time we will go for the cytokinins. And gibberellins again are in general very good for the growth. They help both the shoot culture as well as the root culture, and they have a balanced type of effect on the culture. I would say that they are more of a growth promoter than a regulator. So, these are the 5 hormones which play a very important role in the plant growth culture. Other than this, we can use many types of substances like yeast extract, soya proteins, coconut milk. We can use phenolic compounds like fluoroglycinols. Generally, charcoal is also used at times as a detoxifying agent inside it. Then we have to add a solidifying agent. If we are doing any kind of callus culture, we have to add a solidifying agent, and the preferred solidifying agent is agar. Alginates can also be used, but however alginates are prone to microbial infections. Agar is a preferred one. It does not get hydrolyzed by all kinds of enzymes, and it also helps in maintaining the pH. That is why agar is a suitable agent. So, generally agar is considered to be one of the best type of natural solidifying agent. We have carrageenan, gelatin, starch, silica gel, hydroxyethyl cellulose and polyacrylamide, which can also be used as the solidifying agents. We can use antibiotics to avoid any kind of infections in the medium. Generally, carbonicillin as well as augmentin can be used as the antibiotics. We have to maintain the pH of the medium. So, with this we end the different types of nutritional requirements of the plant tissue culture. In coming lectures, we will be studying further about plant tissue culture. Thank you very much.